I'm going to go ahead and get started with some introductions and some housekeeping, and people will continue to join us, I'm sure, throughout this little introductory spiel. Hi, everyone. Welcome to our second online community education program this spring, Botany for Beginners with Caprice Disbrow. Feel free to say hello in the chat box and let us know where you're tuning in from today if you've just joined. My name is Allison Titus, and I am the Community Education Manager here at the Laguna Foundation. In addition to Caprice, we are also joined by Christine Fontaine, our wonderful Education Director here at the Laguna Foundation. She'll be managing the chat, fielding questions, and adding helpful links to resources while we go through the webinar. Hi, Christine.
Hi, Allison. Hello, everyone. Thanks for supporting our programs, and it's great to have you here with us.
The Laguna Foundation is a nonprofit organization based in Santa Rosa that works to restore, conserve, and inspire public appreciation for the Laguna de Santa Rosa. The Laguna is a wetland complex and a 22-mile-long channel, as well as an entire watershed. If you live in Cotati, Rohnert Park, Santa Rosa, or Windsor, you live within the Laguna watershed. The Laguna is designated as a wetland of international importance, and that's because it is home to an amazingly diverse array of mammals, reptiles, amphibians, insects, and, of course, plants. It's a wonderful place to dive into the practice and study of botany, our subject for today. I'm going to start with a few housekeeping notes for a successful webinar experience. I'm sure some of you are pretty well versed in these at this point, but it bears repeating. You are muted and your video is off, and that is on purpose. If we could see and hear each and every one of you, it would be pretty chaotic. We now have over 100 folks that have joined us for this webinar today. Christine and I will have our video on during the introduction and the closing, and when we take questions throughout the webinar. Caprice will be on throughout the entire webinar. Add your questions throughout the webinar to the chat box and please say hello. You can hover your mouse over the bottom of the Zoom screen and the option to select the chat box should pop up. We will pause a couple times during the webinar to take questions that are coming up in the chat. We will also follow up with an email after the webinar has concluded, answering any questions we didn't get to. All right. Our presenter today is Caprice Disbrow, who just finished an online lecture to her junior college class earlier this morning. Caprice is a professional botanist and instructor of botany at Santa Rosa Junior College and Napa Valley College. She holds a bachelor's degree in plant biology from UC Davis, and her master's degree is in biology from Sonoma State. Caprice is passionate about the diversity and evolution of California native plants, and she serves on the board of the California Native Plant Society Milo Baker chapter, where she edits the newsletter, serves on the scholarship committee, and helps with the education, outreach, and communications. She enjoys teaching people about biology and ecology and strives to make education accessible to all people. This program with Caprice is the first in a week-long celebration of gardening and plants. So stay tuned at the end of the program for more learning opportunities coming up in just a couple days. I think that's all that I've got to start us off. Caprice, I'll hand it over to you to get started.
Thank you so much, Allison. Thank you all for tuning in. I'm really excited to be here to share with you a little bit of botany, botany for beginners. We're going to scratch the surface. We're going to cover some things that you have probably heard of before, and we'll also probably cover some topics that you may have never heard of before. We're mostly focusing today on the anatomy of angiosperms. Anatomy can refer to the organization of cells and tissues within a plant, but that's not what we're focusing on today. We're focusing on macroanatomy, the larger structures that we see when we look at a plant or a flower. And we're looking at the anatomy of angiosperms. Angiosperms are flowering plants. This is a group of plants that have flowers and fruits. Groups we will not be talking about today are conifers, ferns, or mosses. We can do that another time. So today we're going to talk about why should we describe plants? Why should we learn about their anatomy and make these deeper observations? And why should we attempt to identify them for ourselves? Then we'll get into vegetative and reproductive structures. That'll be the meat of our talk today. And lastly, we'll finish with a brief introduction, just a brief introduction into plant identification, what you'll need, and how to use a dichotomous key. So follow along on these slides. Please let us know if you have any issue viewing these right now. So let's start off by thinking about a plant that has intrigued you. Think about your favorite plant. Can you describe it? Maybe you know you love it because of its color or its shape. But let's say you come across a plant that intrigues you. If you don't know the name of it, then you'll either ask someone for the name or you'll have to key it out for yourself. If you key it out for yourself, you'll need to know some of the structures and some of the names of structures to look for to start narrowing down who this plant is, the identity of the plant. But most of you probably already know the name of the plant you're thinking of. Get a good image of this plant in your mind. If you have it in front of you, even better, maybe pull up a picture of it. Because throughout this lecture, I'm going to ask you to refer back to this plant you're thinking of and start applying some of these new structures, features, and anatomy and say and answer, what does your plant have? How does your plant look? So you likely already know the name of it. Why go deeper? Why dig deeper? You may want to talk or write about this plant more specifically, or describe it to someone who's never seen it before. One major benefit to knowing more about your plant is to start making connections between patterns in different lineages of plants. Once you start being able to describe and identify some of these big picture differences, you can apply this to a new plant that you've never seen before, but looks similar to one that you've already seen. So you're applying knowledge that you've already learned from a plant, structures, features, and patterns. And this can help you narrow down the identity of this other plant. Ultimately, learning more about some of these features and organization of these plants helps us be able to identify them for ourselves and to be able to use a dichotomous key. Here's an example of why we might want to take it a little bit further. You may have been taught to identify a plant just on look, and you learned its name. But in this case, where I was conducting a survey in northeastern California, where it's basically the Great Basin, there are two shrubs that are somewhat related and look very similar, but there are some distinct differences. And my client really needed to know the relative abundance of one of these rabbit bushes versus the other. And in this photo, they're growing right next to each other. And you might really be able to tell the difference in foliage color here, but that's not always very helpful. Sometimes the bluer plant doesn't look as blue and the greener plant doesn't look as green. And sometimes folks are colorblind as well or have visual impairments. The reason my client needed to know the difference, and the reason why I needed to look much more closely at these two plants, is because one of these two rabbit bushes is the host to a sagebush checker spot butterfly. This butterfly might use the other shrub as well, but it is very well known to use chrysothamnus for its larval host. And so this client needed to know how much we had of this shrub relative to the other. This was a case where I needed to look very closely at the flowers, the reproductive structures here, to be able to tell the two apart, especially when I couldn't use the foliage as a way to tell the difference. So here's one application of diving deeper, looking at the reproductive structures. So I mentioned that these are closely related, but not exactly the same. You can see that their names are different. And before we get into the anatomy of plants, I want to touch on our classification system. This is a really valuable tool that helps us begin to recognize these patterns and can help us break living organisms into similar groups and be able to identify an individual. So you might have heard of kingdom, phylum, class, order, family, genus, species. Or maybe you haven't, but here they are. These are the major categories of living organisms. Today, we're only looking at one kingdom, Plantae. These are photosynthetic organisms. Today, we're also only looking at one phylum, Anthophyta. These are the flowering plants. The angiosperms is another term for that. Today in this lecture, we'll cover how to determine what class your plant might be in. Remember, I asked you to recall a plant. We'll learn about making sure we know which class it's in. Is it a dicot or monocot? And for these successive subgroups here on out, today's talk will help you be able to identify some of the features that are similar in these groups. This isn't an introduction to plant families, so I'm not going into specifics on each family, but I will touch on characteristics you'll need to be able to recognize and observe if you are to be able to determine which order or family or genus or even the identity of the plant is. And so in this case, for Rosa woodsii, it's in the order Rosales. Nearly every plant in this order have this specific structure. Don't worry if you don't know what a hypanthium is. Then every plant in Rosaceae in this family has a unique characteristic, and so on and so forth. I want to touch on, in the plant world, we use the scientific name. I want to touch on common name versus scientific name. Many of you in the audience may be birders. And in the birding world, common names are not so ambiguous. In the birding world, there is a system for giving a common name. And so it's not ambiguous. But in the plant world, common names are incredibly ambiguous. There is no system. You may have two plants with the same name, like my rabbit brush example earlier. You might have one plant that has many different names. And so the scientific name, which is comprised of the genus and the specific epithet, can be really valuable, not only because it's an unambiguous name, but because the genus and specific epithet tell us something about the plant. The genus reveals its evolutionary relationship. If you're looking at Rosa woodsii and Rosa arizonica, immediately you know, without even looking at the plant, that they're closely related because they both have the same genus, Rosa. Whereas if you're looking at Rosa woodsii and Ribes sanguineum, this beautiful plant shown here on the slide, you know immediately they're not as closely related. They're in different genera, and they may have different features, especially in their flowers. The specific epithet is a descriptor. It can honor a person, like Woodsii, which is named after Joseph Woods, likely the person who first identified or found this plant or named this plant. Arizonica can indicate from where this plant was first found or where it's commonly found. And sanguineum, like this beautiful flowering currant here, indicates red coloration. This flower has a red pigment giving it this pink color. So there's value in using a scientific name. In this talk, I will use both the common name and scientific name for plants that I'll be referencing. But ultimately, what we're going to do today is start looking at some of the anatomy and features that will help us start making sense of these lineages and patterns in different plants. And along the way, as you start to maybe key out plants or make these observations, you'll see that it fits beautifully into the classification system that I just taught you about. So at this point, before we move on, Christine, if there are any big questions, I'd love to answer those right now.
Okay, there aren't any big questions yet, Caprice.
Okay.
There is one question that started out early on. Are angiosperms ferns?
They are not. Ferns are a different group of photosynthetic plants. Ferns do have vascular tissue, which makes them very special in the evolution of plants, but they do not have flowers yet. That would be a fun topic to cover some point in the future. Today we're talking about plants that make flowers and make fruits.
Okay, very good. That was the only question so far.
Excellent. So this is the bulk of the talk here. I'm going to start out with the very basics of an overview of the plant. For those of you who are truly beginners and haven't really thought about plants yet, we're going to start at the beginning. We'll talk about some things that make plants unique and some categories that you might need to know when you start to narrow down what it is that you're looking at and aspects that you can just appreciate nevertheless. And then we'll really get into the floral, the reproductive structures. We'll spend a lot of time looking at the reproductive structures. And I'll touch on stems and leaves as well. So, the overview of the plant. You likely already know that there is an above ground portion and a below ground portion, the shoot system and the root system. But this adaptation is huge. When plants first started to leave aquatic systems and begin to colonize terrestrial systems, these organisms had to deal with these two domains and had to evolve very different structures to be able to survive this new environment. They needed to have more rigidity and strength above ground and really focus their photosynthetic efforts there. And below ground, they needed to fight for water and nutrients, compete for these things. So although this is obvious, I want you to take a second and really appreciate this amazing adaptation. And then think about your plant. Have you ever noticed the roots of your plant? Have you ever thought about what its roots might look like? We're not going to touch on root systems very much today in this lecture. But what I can tell you is that in general, root systems are not as complex as shoot systems. We may have a taproot or fibrous roots, and we may have lateral roots. But that's about it. There are some exceptions, of course. Today, we will touch on some underground stems and stem modifications and some storage structures when we get to stems. But we won't talk much about roots here, except for the difference between monocots and eudicots in just a second. We'll mostly focus on above ground parts of the plant. When you're identifying a plant in the field, if you're not sure if it's rare or not, You shouldn't pull it up. You should not dig up and potentially kill a plant that's rare. And so oftentimes you'll very much begin by looking at the above ground parts of the plant, and you'll primarily look at the flowers. So that's what we're going to focus on today. One big category when thinking about plants is their growth habit. In many keys and floras, sometimes one of the first things they'll ask is your plant an annual or a perennial? This can narrow down the identity of the plant in a big way. And if you're not sure what this means, we're going to cover it right now. An annual plant lives and dies in one single season. A seed will germinate, maybe in the spring. It will grow, and it will, towards the end of spring, maybe summer, fall, it will begin to put everything it has into flowers, and then everything it has into fruits, and then everything it has into seeds, and then it dies. That is an annual plant. The next season, some of those seeds will germinate and the process will happen again. The season can differ based on where you are on the planet. So growth, flowering, and fruiting all happen in one season. And some really nice examples of these here in Sonoma County are the bicolored lupin and the aquatic buttercup, Ranunculus lobii and Lupinus bicolor. So is your plant an annual? It's important to note that when I'm talking about narrowing down these big patterns and these lineages, some lineages have quite a few of both annual and other types of growth habits. For example, there are many lupins here in our county that are annual and many that are also perennial. So this growth habit may not eliminate an entire category when we're narrowing down what our plant is. If your plant doesn't seem to fit this description of an annual, it might be a perennial. Perennial plants live more than a single year. They will grow, flower, and fruit year after year. Some really obvious examples are shrubs and trees. Some not so obvious examples are some small herbaceous perennials. For example, the fairy slipper found in our forests, our mixed evergreen and mixed coniferous forests here in the county, is a perennial. This grows from a bulb, a below ground storage structure, modified stem and leaves, that for most of the year, this plant does not have any tissue above ground, but yet it comes back year after year. This is a perennial plant. The Western dog violet is another example of a fragile herbaceous perennial that comes back year after year. Another great example are our California poppies. So you may already know this, but is that plant that you were thinking of? Do you know right away if it's a perennial or annual? You may need to observe your plant in nature throughout a season or two to get a feel for this. Another category are biennials. I don't have a slide here for that because we don't have as many great examples here in California. In fact, if you search biennial in CalFlora, a great online resource for plant identification, not many plants show up, although we do have some. Biennials grow vegetatively in their first season and then flower and fruit in the second season and then die. They live for 2 seasons typically. So growth habit, annual or perennial. Another big category your key or flora might require you to know the difference of is it a monocot or eudicot? So let's talk about what is this cot, monocot, eudicot. This has to do with the cotyledon. The seed leaf is another term for cotyledon. In one big class of plants, the monocots, their embryo and little germinating plant, when the seed first germinates, will have one seed leaf, one cotyledon. This leaf-like structure will provide nutrients to this young plant early on. Eudicots, true dicot for two cotyledons, will have two. This image over here might be one of the, I think it's the only non-native plant in this talk, but I couldn't help myself because its cotyledon is just so adorable. But you'll see here that its cotyledon looks much different than the true leaf of the plant. This plant had two of these little heart-shaped cotyledons. This plant was a dicot. Is your plant a monocot or eudicot? Some of the best ways to tell are by looking at your leaf venation. If your veins, the ridges on your leaf, really the plumbing of the plant, if your veins are parallel, then it's likely a monocot. But be careful, some leaves have very prominent veins that look parallel, but there'll be reticulate net-like branching veins in between. So look closely at the leaf of your plant to look for this. If you have branching, reticulate, net-like venation, you might be looking at a eudicot. Another huge way to tell the difference is by looking at your floral parts. Monocots are typically monocots typically have floral parts in threes, 3, 6, 9, 3 petals, 3 sepals. We'll get into those terms shortly. Whereas eudicots will have floral parts in 4s or 5s, or maybe many, maybe 11 or more in some cases. There's actually a third group of angiosperms, of flowering plants, that we call the basal angiosperms, the most ancient early flowers that actually look a little different than monocot and eudicot. If your plant that you're thinking of doesn't quite meet either of these, you might be looking at a basal angiosperm. This is a whole nother topic, talking about the evolution of our early angiosperms to our most derived angiosperms. But for the sake of this class, I'm hoping that your plant fits into one of the two, the monocots or the eudicots. So before we really dive into reproductive structures, do you have any questions?
Yes, Caprice, there's one question about California poppies. Are they annuals or perennials?
They are perennials. You might find these plants with very thick kind of base of their stem and roots. And you might find that these plants regrow in the same location year after year. It is possible to treat poppies, California poppies, the Schultzia californica, as an annual, depending on maybe your gardening methods. But they often, in coastal settings, will grow a very thick, put a lot of energy and investment into a good root system, and will grow back year after year.
Okay, thank you. Do we have time for another one?
Sure.
Okay, this was a question about planting an annual garden. So if you're going to plant an annual garden, is it best to kind of let the seeds germinate on their own year after year? Or do you need to directly reseed each year?
That will depend on your maybe non-native invasive seed bank that might be outcompeting your annuals if left alone. Some methods I've heard of are to plant your annual seeds in rows. That way you can weed non-native plants in between. And this will give those annual seeds a chance to grow and mature and make a lot of seeds. And there's a potential that the following year and year after that, that you'll have a nice amount of those native seeds. However, if you have a lot of Bromus diandrus or Avena barbata, these ripgut brome or wild oat, these grasses that grow tall, fast, lay down a lot of thatch. It can be difficult to let those annuals for them to come back and compete with these other plants. So it depends on your setting. Feel free to email me with specifics and I can maybe have some other techniques that can help.
Okay, thanks. And one other general question, are eudicots the same as dicots?
Great question. Back in the day, researchers, folks looking at plants, tried to put everything into monocots or dicots. And things weren't quite fitting. And that's because of those basal angiosperms. Early flowers, these most ancient flowers, have some characteristics of what we now call eudicots, but also have some characteristics that don't fit either category or that actually align with the monocots. So we were troubled with this categorization until we realized that those are the more ancient flowers. And here's all the reasons why they're more ancient. We can see it in the record, in our fossil record, and we can see it in the evolution of their structures. And then later on, the monocots formed and the eudicots, true dicots, the more advanced angiosperms were categorized. So that's why you may see dicot or eudicot. We typically in curriculum don't use the term dicot anymore. It's basal angiosperms, monocots, and eudicots.
Great, thanks for that clarification.
No problem.
Okay.
So parts of a flower. We're going to start off with the general model of a flower. Just cover those terms, some of which you likely know. Some might be a little fuzzy or new. We're going to get into some variations of flowers. And we're going to cover some variations in individuals based on those flowers. We're going to talk about where those flower parts join up in relative to the ovary. This is a very defining feature that can help you key your plant, determine which lineage it's from. We'll talk about union, then fusion of parts, and then we'll take a little bit of time to look a little bit more closely at some special examples for each of these parts of a flower. You've likely seen a figure like this before, but let's make sure we're all on the same page. Starting at the bottom, the base of your flower is called a receptacle. Think of your plant. What does its receptacle look like? Have you ever looked at it? I can tell you this, the California poppy has a really cool looking receptacle. Have you ever noticed it? Next, we'll have typically, but not always as I'll get to shortly, a layer or a whorl, a layer of structures called sepals. Following the sepals, more inside from those are our petals. And then typically inside of the petals, we'll have our male structures. This will be an anther and a filament. In our more advanced angiosperms, in our more primitive ones, this might look a little different, but typically still will be inside the petals. And together we call the male structure the stamen. Anther, I have a way of remembering that this is male. My partner, his name is Anthony, he identifies as male, and so that helps me remember that anther is a male structure. And stamen has the word men in it, so that can help you recall that these are male structures. And then typically in the very inside of a flower, we have our female portion of the flower. In a couple slides here, I'll talk about carpels versus pistils. But when talking about the entire female structure, I'll be using the term pistil, and I'll tell you why in a little bit. The pistil is comprised of the stigma, where the pollen lands and is received, the style, the tunnel through which that pollen needs to dig, the ovary, which is what turns into the fruit, and the ovules, which will become seeds. So all flowering plants make fruits, and it's because they have this unique structure called an ovary that will swell and change to develop into the fruit. These are unique aspects of angiosperms of flowering plants. So this is just a general model of a flower. The flower that you are thinking of or holding might look totally different, but you should still be able to see some of these layers, the sepals, petals, stamens, and pistils somewhere in your flower. And we'll talk about some modifications moving forward. Not all flowers have all four parts. You may have seen or heard of this before, so we're going to put some names to this. There are perfect and imperfect flowers. These imperfect flowers are no less amazing, despite the name. A perfect flower contains both the male and female parts. Imperfect flowers lack either a male or female part. Is your flower perfect or imperfect? This will help you if you're trying to key out or figure out who your plant is. Similarly, but different, is complete versus incomplete. A complete flower has all four layers: sepals, petals, stamens, pistil. Incomplete is lacking any one of these. It could be lacking sepals, it could be lacking petals, it could be lacking one of the reproductive parts, which would make it imperfect as well. So you can kind of have a couple different combinations here. But these are things you'll want to look for. Maybe your favorite plant is beautiful, you've seen it a million times, but maybe it's actually lacking one of these layers. Have you ever looked that closely at it? A really great example is the California hazelnut. This is a beautiful shrub in our understory of our mixed evergreen forests here. The leaves are so soft, they're lovely. But did you know that it has imperfect flowers? It has a female-only flower, and it has male-only clusters of flowers that produce a lot of pollen. This is a really nice example of an imperfect native plant here in California. So a little bit more on imperfect flowers. If your plant has separate male and female flowers, but they're on the same plant, then we call that monoecious. Hazelnut is a monoecious plant. Although the female parts and male parts are separate, they occur on the same individual. Alders, oak, these are examples of monoecious plants that have imperfect flowers. Dioecious, however, means that the individual only has female flowers or only has male flowers. And then wonderful example of this is coyote brush, Baccharis pileolaris. How many times have you hiked by Baccharis or coyote brush and noticed that it's in flower, or maybe it kind of has a distinct scent, not very strong, but a unique Sonoma County shrub? Have you ever noticed that the flowers are different? Well, here you are. The female flowers have a little bit more of this fluffy pappus. The male flowers a little bit more dense with their stamens. You might notice that there will be male shrubs and female shrubs nearby. They're depending on pollinators to transfer that pollen from one shrub to the next. These are dioecious. It's another strategy in the evolution of plants that works but it's something to begin to notice if you want to start understanding plant lineages and be able to identify them further. Another big characteristic of a flower that you may or may not have ever thought about is, where does that ovary sit? So recall, the ovary is what houses your ovules. The ovary will become the fruit. Your ovules will become the seeds. Different lineages of flowers have a different placement of the ovary. We call this the insertion. Where are the petals attached? Where are the sepals and stamens attached relative to your ovary? So you might have a hypogynous flower, meaning below. The insertion of your petals and stamens are below your ovary. You might have a epigynous flower, where the insertion of the petals and stamens and sepals are above your ovary. And there are different variations of perigynous. Peri meaning around, like perimeter, where maybe it's forming a fusion and a cup, and it's kind of partway. Maybe it is actually fused to the ovule, but it's still halfway. Your flower, is it hypogynous, perigynous, or epigynous? You might also hear superior or inferior. So a hypogynous flower has a superior ovary, and an epigynous flower has an inferior ovary, meaning inferior meaning under, superior meaning over. These are some features that will help you narrow down your plant and who your plant might be and who your plant might be related to. A really fun example, although I don't have a photo here, are some of the plants in the family Ombraceae, such as primroses, or our sun cup that you might see along our coast. This plant, this beautiful yellow flower grown in coastal grasslands, has what looks like a long stem to the flower. And if you start to dissect that flower, you'll have trouble finding its ovary, because it's not actually, it is inferior, but it's nowhere near the flower. It's actually pretty far away in the base of the plant, very protected. And if we think about the evolution of plants, superior hypogynous ovaries are more ancient and inferior ovaries are more advanced, a little more protected. So we're diving a little bit deeper now into female parts specifically. I mentioned this carpal pistil, these terms for the female parts. And we're getting into the nitty-gritty, but for a good reason. If you want to start understanding your plant better, you might want to look at that female part more closely. In this figure, I have a single unfused carpel, also called a simple pistil. In this example, we have multiple unfused carpels. Each are a simple pistil. This is relatively uncommon. We see it in our more primitive plants, like our buttercups, like ranunculus. We'll see very separate carpels and therefore separate pistils. But more often than not, there is fusion of carpels. And we call the entire structure our pistil. This is most common. And so I typically call the female part the pistil. And there can be clues to how many carpels are fused. Maybe the stigma is branched. In this case, we see 3 branches. That tells us there's likely 3 fused carpels. But we might also just see lobing of the stigma. And that can be a hint to how many carpels are fused together, creating this female part. Within each carpel, we'll have ovules attached somewhere inside. But if you've looked at a fruit of any kind, you'll know that there's great diversity in where those seeds are found and where they're attached. Have you ever sliced into a flower and looked at how many cavities there are in the female part? Have you ever sliced into the female part and noticed where those ovules are attached? They're not always attached in the center, like shown here. They can be attached along the far edges. They can be attached partway through. Even though there's 3 fused carpels, sometimes these membranes are present or not. So I encourage you, the next time you're looking at your favorite plant or flower, make a cross section of that female part and see if you can determine how many chambers, how many compartments, and notice where those ovules are placed. These are questions a key might ask you regarding your plant. Okay, so this is a bit more on the female part. Do you know right now, does your flower have a branched or lobed stigma? Do you have any clues, without cutting into it, how many chambers you might have? Maybe you're familiar with the fruit and how the fruit breaks open or looks when you cut into it. That can tell you a bit about what has happened here in the female parts of the plant. Now let's take a second to look a little bit more at the male parts, the stamens. There are lots of modifications to stamens, but today I'm just going to show you 2 examples of modifications. If you look at any glossary, you'll see there's many terms for different setups of stamens, and I'll tell you about 2 of them today. In the family Malvaceae, the mallow family, we have stamens that are fused together, more or less. This is called monadelphous, one brother, and it's really prevalent in this group of plants. This is one of those examples where you can see fusion of male parts and know pretty quickly, I might start looking at Malvaceae because I've learned that this pattern is common in this family. Fusion of the male parts. We have the female part right in the center, but the male parts are fused at least partially. This is California Fremontia, a beautiful shrub native to this region. And this is a Checker mallow, another Malvaceae beautiful flower with monadelphous stamens. Another modification to male parts, but definitely not the only other, there are many others, but another one that's very common to mustards or Brassicaceae, the mustard family, is this tetradynamis. Let me tell you what that means. There are 2 sets of stamens. One set has 4 long stamens, and one set has 2 short stamens. So in this group, you'll often see 6 stamens, 4 that are long and 2 that are short. We can see it a little bit better in this flower here, looking at the milkmaids, one of the first flowers to show in early spring. We've got 1, 2, 3, 4 long ones and 2 short ones. This beautiful flower, Coast Rock Crest, is a flower I had the honor of identifying during a survey for a client. And this plant is CNPS ranked 4.3, meaning it has limited distribution, which is the 4, and the 0.3 means it's not very threatened. Only about 20% of the occurrences are threatened. So although this isn't a bit federally protected or state protected like CNPS 1 or 2 category, it's still valuable to know that this plant has some kind of rare or threatened ranking. And so I was happy to find this little beauty and inform the client of its presence. So these are just 2 examples of modifications to male plants, to male parts. Sepals can also vary. They can be present, they can be fused, they can be free, they can be modified greatly, and they can also be deciduous where they fall off. So does your plant have a unique set of sepals? And lastly, we'll touch on flower petals here. Altogether, we call the petals the corolla. These can be free, they can be fused into bells, into cylinders, but something you might want to notice is the symmetry of your flower. Is it radially symmetrical or is it bilaterally symmetrical like we see here in this penstemon or in this buttercup? Flower corollas and petals can have other structures like spurs or glands as well. These are things you'll want to take note of. So we are beginning to run out of time, but that's okay because most of what we've just talked about, these parts of floral structures, are primarily what you would need to begin plant identification. So I've skipped over our vegetative leaf and shoot structures. Feel free to email me if you'd like to see more about that. But I'd like to use our last minute or two to introduce you to how you might use these new terms for flowers, for reproductive parts, to key out a plant. You'll need a key, a flora, and maybe a glossary of terms, because there's many terms, a dissecting scope or hand lens, and maybe some manipulation tools, forceps, and a ruler. And what you'll encounter is a dichotomous key. A dichotomous key gives you 2 options. Dicot, di for 2, dichotomous key, a couplet. And you'll choose the option that fits your organism more closely. So if we're looking at Jolly Ranchers, for example, is it chewy or is it hard? It's a hard candy, right? If we move over here, this leads us to number 7, to couplet number 7. That means we'll skip couplet 2, couplet 3, couplet 4, couplet 5, couplet 6, and we'll move all the way to couplet 7. And we'll choose the option that fits our candy better. Is a Jolly Rancher more spherical or not so spherical? It's not so spherical, it's not ball shaped. So we'll move on to couplet number 9. And we'll look at 9A or 9B. Wrapper transparent or wrapper tells the flavor? The wrapper does tell the flavor. We can follow that over and it tells us what we're looking at. So now that you know some of the terms that you'll encounter in a key when you're encountering these couplets, you might be able to key out a plant for yourself. So thank you so much for tuning in today. I don't think we'll have much time for questions, but feel free to email me and we'll also be responding to some of the ones in the chat as well. So thank you so much for tuning in.
Thank you, Caprice. All right, if there's an easy, are there any easy questions out there in the chat, Chris, that we want to address before I give my closing announcements?
I don't think so. Most of them were answered.
Okay, great. We will follow up with an email with answers to some of those questions in the chat, so stay tuned for that coming from Eventbrite. Thank you so much, Caprice, for sharing your expertise and your passion and for opening our eyes to the wondrous and complex world of botany. I think it's a great study to pick up while you're sheltered in place. And thank you all so much for attending today and supporting our online programs. If you have the means to donate at this time to support our critical conservation, restoration, and education work in the Laguna, we would really appreciate it. And thank you to those of you who donated upon registering for this program. As I mentioned before, this program was the first in a series of online programs leading up to the virtual eco-friendly garden tour on May 2nd. I'm going to list some of those other events that are coming up this week that you can participate in. CNPS Milo Baker chapter, which we've mentioned a couple times in this webinar, is having an online native plant sale right now. The plants you purchased help support the education and stewardship work of the chapter throughout the year. You can check out their website to purchase plants, which will be delivered directly to your door between May 2nd and May 6th. However, I'd hurry to check it out if you're interested because many of their plants are already sold out. There's a very limited supply, so you can check that out ASAP, I would say. On Thursday this week, we will be hosting a Wildscaping for Native Songbirds webinar with Veronica Bowers, the founder and director of Native Songbird Care. That's at 2:00 p.m. on Thursday. The virtual eco-friendly garden tour goes live on May 2nd. You can watch videos from gardens around Sonoma County that showcase water-saving practices and beautiful plants. After watching the videos, you can join the interactive Q&A session that we are co-hosting with Dailyaks at 4:00 p.m. to get your gardening questions answered live. You can also check out our Laguna Foundation community education page for other online events coming up in May. We are uploading new events all the time, so check back often or subscribe for our e-newsletter where you'll get an events-only email each month. All right. Thank you so much for tuning in. This talk was originally going to be a part of the in-person eco-friendly garden tour, so thank you, Caprice, for your flexibility. I think that's it for my announcements. I hope you all out there in the audience take good care of yourselves, and thank you again. See you next time.